Thank you very much, Audrey, for this great introduction, and to everybody who is interested in this particular work. So, as you heard about the cerebellum earlier this morning, now we are moving to another brain region, and actually postnatal development. So, what I'm going to talk about to you today is hippocampal neurogenesis, and specifically what is it? What is it for? What stimulates it? What suppresses it? And what is the role of fatty acids in neural stem cells, actually the metabolism? So finally, how might a biomarker for neurogenesis be used, which might be very useful in studying certain pathologies in your clinic? So the question whether we have neurogenesis after birth has been plugged by almost 100-year ascertainment by the father of neuroanatomy, Ramon y Cajal, who made the first brain atlases and observed that in the adult, the nerve paths are something fixed, everything may die, nothing may be regenerated. So it was no wonder that, when we know that there is a lot of postnatal development for myelination, synaptic maturation, activity-dependent morphogenesis, and so on and so forth. But it is only for the past 20 years that we have, in a way, ascertained that there is also neurogenesis happening after birth. However, this discovery was very turbulent, and it has this rather interesting history that I wanted to point to you. So when the first report of newborn neurons in the rodent hippocampus was presented in the 1960s, they used then a very new technology, radioactive thymidine labeling. So this radioactive substance gets incorporated into the dividing DNA. And using this technique, at that time, Altman and Das saw that there are newborn neurons in the hippocampus. Now, this technique really was very new at that time, but very quickly later on, actually 20 years later, this is now what we use regularly in our labs. Everybody who does cancer studies use the same technique to label proliferating cells, and this analog is called BRDU. However, because of this novelty of the technique used, the work of Altman and Das was completely discredited and attributed to an artifact. So Altman lost his tenure. He moved to a small university and eventually disappeared from the neuroscience field. 10 years later, in the 70s, another set of investigators used electron microscopy to prove his findings. And they were also discredited by the work of Paško Rakić. So Kaplan actually moved to pharmaceutical industry, and then again, they all sort of move away from academic career. It was only in the 90s that this notion of adult neurogenesis in the hippocampus started to be ascertained in first mouse models, and then a little bit later on in people, with seminal work from Peter Eriksson, and then followed by Christy Spalding. So we thought, now we know we have adult human neurogenesis in the hippocampus. And then in 2018, a paper from UCSF came and said, well, we cannot see it, therefore it doesn't exist. And these negative findings really, were far stronger than all positive findings that we have, including the paper that came a month after this sort of damning Sorrell's paper. And later on with studies in Alzheimer's disease and aging and so on and so forth, we are still struggling in this rather turbulent field to know whether humans really have this neurogenesis. Now, from rodent studies, that nobody disputes. We know that stem cells reside in the subgranular zone of the dentate gyrus in the hippocampus, and there newborn neurons are made every day. So these newborn neurons are very important for something we call pattern separation, which is the way to distinguish two different objects. So, for example, if you see this apple, you will know it's a certain kind of an apple. And when you see this one, you know it is also an apple, but it is not the same. It's similar. However, people who have depression, people who have some cognitive, intellectual disability, cognitive impairment, neurodegenerative diseases, they really cannot distinguish these two objects as similar. They sort of see them as the same. So, for example, the same would be if you have a set of keys, you know instantaneously which keys are for your home, for your post box, for your car. But people who have certain disorders associated with decrease in neurogenesis, they will need to think, which one of these keys really is different. And that is because they have this poor pattern separation and they cannot distinguish subtle differences in the patterns. So we also know that this is linked to neurogenesis by seminal work from Craig Stark. So he designed this so-called Stark test, where you present the participants different sets of stimuli, and you can see here that some of them are similar and some of them are completely different. So the participant needs to identify new, old, or similar. And this was linked by to hippocampal neurogenesis in the dentate gyrus by fMRI studies from Craig Stark lab, where he could see that it highlights this task, particularly highlights this region. So now we also know that there are other stimuli that can increase neurogenesis, running in particular. Cognitive stimulation, so enriched environment, really stimulates production of new neurons. Also, therapies that are designed to treat depression, so antidepressants, deep brain stimulation, electroconvulsive shock, they're very robust stimuli of neurogenesis. We also know that aging and epilepsy in particular lead to transformation of these cells to astrocytes. And so during aging and in epilepsy, you have more of gliogenesis rather than neurogenesis. We know that certain circumstances are reduced associated with reduced neurogenesis. So chronic illness, depression, addiction, schizophrenia, autism, but also chronic stress, chemotherapy, radiation therapy, social isolation, they really cause the decrease in neural stem cell population and neurogenesis. So given all that, and the fact that the seminal carbon dating studies calculated that about 700 newborn neurons are integrated into the human hippocampus per day. So 700 on the left and 700 on the right replace about 30% of the dentate gyrus over the human lifespan. So as you're hearing all these talks today, because for you, this is something totally novel, so it's considered cognitive stimulation, enriched environment. So you can just imagine that while you're learning all this information, the newborn neurons in your brain are really integrated in making new memories of what you have just learned. So given all that, there is a very high interest to utilize this process and target it as a therapeutic means to sort of alleviate disorders that are associated with decline in neurogenesis. So we have been interested in this targeted modulation of neurogenesis and not only working on the basic science level, what is the biology of these stem cells, how can we modulate them in a targeted way, but also how can we detect this process in the live human brain. because as of now, that has not been possible. So everything that we know is for post-mortem human brain. And can we really quantify the response of neural stem cells and neurogenesis to a variety of stimuli and therapeutic agents that could be developed to modulate neurogenesis? So to make things short, we started to look at metabolism. And we went back to the cancer field, where we knew that all proliferating cells have to make new membrane for the daughter cell. So they depend on lipid metabolism. And for their energy, they also use anaerobic respiration, fatty acid oxidation, and de novo lipogenesis in order to make new cell viable. As these cells progress through the cascade to the mature granule neuron, they lose this dependency on lipid metabolism and actually completely transfer to oxidative phosphorylation, aerobic respiration, dependence on sugar and oxygen to make ATP. So this meant that stem cells that we are very interested in imaging and studying really should be abundant in fatty acids and lipids. So we decided to study them by looking at primary cultures of these different cells using very high field NMR. And then once we defined their isolated profile, to move to MRI imaging of the live animal models, particularly mice. And then once we ascertained those biomarkers, to apply this technology to human brain spectroscopy. And because I'm really interested in early human development, I decided to use human brain spectroscopy in order to really be able to image even prematurely born babies. So to make things short, we started to look at fatty acids at signaling molecules and progenitors, and also to fatty acids at biomarkers in the live human spectroscopy. So we discovered that one acid, oleic acid, binds to a very key molecule in stem cells called TLX. So in the native state, this nuclear receptor TLX is occupied by a co-repressor. The stem cell is quiescent, resting. When the neurogenic stimulus comes, oleic acid enters the nuclei, repels this co-repressor, and recruits a co-activator of this nuclear receptor. It changes its conformation, and it starts to activate downstream genes for neurogenesis. As this happens, new cells are formed. They are releasing more and more lipid signals, and we can pick this up in the human brain spectroscopy in the hippocampus. So there, to do this, we had to design a new analytical algorithm that really isolates this fatty signal from the spectroscopy of the hippocampus. And you can see here how we could really distinguish it from the cortical control region. We did a large study scanning over 400 participants over 8-9 centers throughout the world, and saw the same downstream trajectory over age. And we also saw that children, females, adolescent females with depression have very little neurogenesis happening in their brain. So now, equipped with this knowledge, and we have done a lot of other studies that are also in using SSRIs, electroconvulsive therapy, and autism, and so on and so forth, that I'm not presenting here for the sake of time. So we now think that we have the biomarker that we can use in humans to detect and quantify this process in the hippocampus with really robust accuracy. So now that we have a target, which is this nuclear receptor TLX that is activated with oleic acid and can induce neurogenesis, and we have a biomarker that we can utilize to measure it, we are now moving to designing new drugs to promote neurogenesis. So this is done in collaboration with Dr. Young at Center for Drug Discovery and at Baylor College of Medicine. And there we use two different technologies for drug development. One is so-called fragment-based drug discovery, and the other one is DECTEC, when they link these little small molecules to a DNA code. And so you can really screen your target with million compounds very quickly, and based on this DNA code, you know which ones are active and which ones are not. So we already have certain candidates that we test in our human patient-specific brain organoids. We make them by using skin cell from a patient or from a healthy individual. And in a dish, you can make them into the fibroblasts into induced pluripotent stem cells that then, using certain growth factors and other factors, you can differentiate into something that is commonly called mini brains. So we study these mini brains in a dish, and they're very important for development of personalized precision therapy, because you can really target it to the person who gave you the skin sample. So here you can see that, in these brain organoids, our first drugs that are labeled here in gray and yellow bars, they can induce stem cell and new neuron production in these little brain organoids. And when we give some of these compounds to the mice, because that is where we can really study their kinetics and so on and so forth. So we give them orally 5 days, and then we sacrifice the mouse and give that BRDU analog that I was talking about initially. So we can label the proliferating stem cells and the newborn neurons. So we see that some of these drugs really expand the population of newborn cells in the hippocampus. And here we did some studies of the behavior. So one of these drugs at 10 milligrams per kilogram enhances the working memory that we test in this Y maze when the mouse is traversing a platform that looks like the letter Y. So in any case, you can measure how exploratory and this behavior is, and these neurogenesis drugs really seem to activate this memory. So in summary, I hope you learned a little bit more about this phenomenon that nature gave us to repair and regenerate a very important center for learning and memory and mood control, namely hippocampus. So our work has indicated that we can study this process in the live human brain using spectroscopic biomarker that is specific to neural stem cells. And it's very sensitive to changes during aging and in depression. Now, we also have this new ligand of the nuclear receptor TLX, which is important for increasing neurogenesis. And we are now designing new drugs that mimic the structure of oleic acid, and therefore we can apply them and give them to patients who have problems associated with lack of neurogenesis. Most recent work that I did not present because it's really happening as I speak is that we have discovered patients that have a missense mutation in this particular protein gene. And so they have intellectual disability to a degree. So we are now working with physicians who have these patients, who have reported them, to study them a little bit more using these neuroimaging and cognitive and mental health assessments to see whether our future drugs that we develop targeting this particular mutated protein might help these children learn better. Thank you very much.
Wow, Dr. Savatic, that was amazing. We've got lots of questions for you. One is about whether anybody's looked at people who exercise versus those who don't.
Actually, we are doing the study. So I have a lot of collaborators all around the world. I don't know why the last slide was not there, but this study could not be accomplished without help of so many investigators that we have in Australia, China, Europe, all throughout Europe, and so on and so forth. Because these studies are very expensive, and so I welcome all those collaborations. So the collaborators in Amsterdam are actually looking at soccer players. So the Ajax team, so they're scanning them before their season begins, and they're scanning them after the season to see whether that huge running over the football field, can help increase this signal. But that is one of the sort of scenarios that we are really keen to test in people.
I wonder, yeah. I'm wondering about heading the ball and how that's going to affect neurogenesis in soccer players. So there are some people asking about dementias. Specifically, is there a difference in regional stem cells and new neurons in various dimensions, in various dementias like Alzheimer's, FTD? And could your techniques be brought to bear in studying these issues and perhaps find patterns that may predict future dementias? Lots of
this is an excellent question too. Yes. So the only data that we have from Alzheimer's disease post-mortem studies comes from Spain. And those were some of the seminal studies of neurogenesis in old people and in people who have Alzheimer's disease, different Braak stages of Alzheimer's disease. So what they showed is that we do have less neurogenesis in normal aging, but this really starts to drop with mild cognitive impairment, and particularly in Alzheimer's disease. Then the other study came from Chicago, from University of Chicago, Orly Lazarov's lab, where they used skin cells, the same type of the protocol that I showed you, and they used it from Alzheimer's disease patients. And they developed induced neurons, so patient-specific neurons in a dish. So they showed that the newborn neurons are really made at a slower rate in these cultures. So what we are hoping now, and we are having a collaboration now with Methodist here at Houston, as well as the Spanish group that has really a large cohort of patients for Alzheimer's disease. So we are starting to scan them, and hopefully that is the idea, that we can design a protocol that can be used in primary care. That every 5 years you can go and get your MRI scan, including the spectroscopy, to measure the signal and identify very early on in the live brain, do we need to do something more to prevent decline in neurogenesis and cognitive? What I'm
I think we have time for
just a second. So what I'm hoping is actually to do this in kids who have intellectual disability, who have, certain types of anxieties that are linked to hippocampal function. So we also have a program here to scan prematurely newborn babies. because they have the population of stem cells that is smaller than the population that you have, 12 weeks following, to full term. So to see whether this population that you're born with actually influences learning disability or specific types of learning disability later in life. Maybe anxiety, depression, and so on.
That's so exciting to hear. I think over the next decade, this is going to be a very exciting field to follow. We have a question about indication for therapies for stroke patients.
Oh, stroke is a little bit different. So we envision neurogenesis is important for the spatial learning, pattern separation, things that, and then mood control, which is in a way intuitive. So the happier you are, you learn better. You learn better, you're more happy. So in stroke patients, this could be applied to stimulate. So this is really particularly specific for the hippocampus. What people have been doing for stroke is transplantation of bone marrow-derived progenitors. There is a study, I think, at UT Health Science Center here in Houston that is specifically looking at that therapy to increase, newborn neurons within the stroke area, which is mostly, in the cortical regions, not really in the hippocampus. So those therapies are a little bit different. They're cell-based therapy, not pharmacological-based therapies. But I know that there are lots of places that are trying to develop it for stroke specific.
Great. I have one more question from the audience, and then I'm just going to have to email you all the questions that I have. Okay. So this person is asking, is there any link between epigenetic changes and neurogenesis?
Yes, that is a very large area of study, particularly bisulfite methylation. So we do know that there are epigenetic modifications of neural stem cell per se, and those probably lead to their loss of capacity to divide. As we age, these stem cells change. So when you're young, they can produce a lot more newborn neurons than when we reach adulthood and then older age. So their speed of division decreases. They're far more silent. They're also far more prone to make glia. So as we age, there is increased glia in the dentate gyrus. And that has been attributed to gliogenesis rather than neurogenesis as we age. So we want to prevent that. And so those changes in the stem cell biology are most likely mediated by some epigenome changes in their, milieu of the DNA.
Mm-hmm. Well, thank you so much, Dr. Savich. This was fantastic. And I'm sure lots of people are going to have lots more questions for you at the meeting tomorrow. So next time,
send me those questions.
Wonderful. Thank you.